The aim of this practice is to describe and use the principal genetic engineering laboratory material and equipment in order to know their application and utility. The correct manipulation of a micropipette is, first, to take the sample, you have to adjust the required volume, choose the correct tip, and then press the plunger until the first stop. Introduce vertically the micropipette into the solution and let the plunger button take the sample slowly, avoiding air in the tip and drops of the sample surrounding the tip. To release the sample, you can touch the wall of the tube or the microtube or get in contact in the accepting solution. Then you have to push the plunger until the first stop slowly, and without releasing, you go all the way until the second stop. Still pushing down the plunger, you take out the micropipette on the solution and eject the tip with the ejector button in an appropriate jar.
In order to learn and understand how properly use the micropipette, we did an exercise where we used 3 different micropipettes. One of 20 microliters, another one of 200 microliters, and a third one of 1000 microliters. In each one of these, we took 10 samples of deionized water using the lowest, the middle, and the highest volumetric capability. To make sure the results were accurate, we weighed them in an analytical balance. To weigh the samples, we used a petri dish cover, tearing each time we added a new sample, and registered the results in a chart. Then we measured the standard deviation and the mean of the 3 micropipettes.
For the last exercise, we took several times the same volume of distilled water and placed it in a microtube. With the 1000 microliters micropipette, the sample was taken 3 times by adding each time 330 microliters for the tube to have 990 microliters, which then were weighted. The 200 microliters micropipette was also used thrice to take 48 microliters in order to get 144 microliters. The 20 microliters micropipette was required 4 times, taking 3.5 microliters to obtain 14 microliters. Each of the following steps were repeated 3 times, and the results were recorded in a chart. This exercise was performed to make the students aware of one way to measure volumes that are near the lowest or the highest boundary of a micropipette.
In conclusion, we can assume that it is very important to have a correct manipulation of the micropipette in order to avoid making mistakes of measurement as a consequence of air inside the tip, not taking the exact amount of solution, or equipment misusage.